When you titrate an acid with a base, the trace of pH versus volume of base added gets a characteristic S shape. Why is that? Well, first of all, let's talk about molarity and normality. The normality of a solution is the molarity multiplied by the number of protons the acid gives off, or the number of protons the base takes in. For example, 1 molar hydrochloric acid is also 1 normal hydrochloric because hydrochloric acid is monoprotic. But 1 molar sulfuric acid is 2 normal because each mole of sulfuric acid gives off 2 moles of protons. This is a sample titration apparatus. A solution of known normality is in the burette, and at the end point of the reaction, or the equivalence point we should say, the volume used is recorded. Here in the Erlenmeyer flask you have a solution of known volume but unknown normality or molarity, and that's what you're going to titrate into. Now we assume that a strong or weak acid is in the flask, and we want to find its concentration by the titration. We would titrate with a strong base, typically sodium hydroxide, of known normality or molarity. Let's look at stage 0. We have added no base. We've got 0.01 molar hydrochloric acid in the flask, and there's 25 milliliters of it there. We've added no sodium hydroxide. The only thing we've got is 0.25 millimoles of hydrochloric acid in the flask. The pH then is the negative log of 10 to the negative 2, which is 2. Stage 1, we add 10 milliliters of the base. That's 0.1 millimoles of sodium hydroxide. It neutralizes 0.1 millimoles of hydrochloric acid. We then subtract that from the original 0.25 millimoles of acid in the flask, and we get 0.15 millimoles of hydrochloric acid. The new volume is 35 milliliters, so the concentration, we divide the millimoles of acid by the volume, and we get 0.0043 molar. Plug that into your calculator, and you see that the pH has gone up to 2.37 from 2. In stage 2, we add another 10 milliliters of base. That's 0.1 millimoles of sodium hydroxide, neutralizes the next 0.1 millimoles of the acid, leaving, as you can see from the calculation, 0.05 millimoles of acid, and now we have 45 milliliters of this diluted acid. Concentration 0.05 over 45 milliliters is 0.0011 molar. The pH is now 2.95. Stage 2.9 we'll call it. We're going to add 4 more milliliters of the sodium hydroxide, and that's 0.04 millimoles of sodium hydroxide. This neutralizes the next 0.04 millimoles of the acid, leaving only 0.01 millimoles of hydrochloric acid in 49 milliliters. The concentration is 0.01 over 49 milliliters, 0.0002, or 2 times 10 to the negative 4th. The pH has only gone up to 3.69. Originally, remember, it was 2. We've added 24 milliliters of the base, and we're only up 169 basis points on the pH. At the end point, we'll call this the end point and equivalence point the same, we add the last milliliter that we need to neutralize the last millimole, 0.01 millimoles of the hydrochloric acid. That leaves no millimoles of hydrochloric acid in the flask. We only have the hydrogen ions that are contributed by the autoionization of water. The volume is 50 milliliters with no excess acid or base. We just have salt water sodium chloride. Therefore, the pH must be 7 because this is a neutral salt that's left. You see that sharp upturn in the pH that's taken place here in about 4 pH units, roughly 3 or 4 pH units with just 1 milliliter. Now at stage 3.1 we're going to add 1 milliliter of the base. There is now no more acid to neutralize, so that base is free in 51 milliliters. We have 0.01 millimoles of sodium hydroxide in 51 milliliters. We're now calculating the hydroxide concentration, which is 2 times 10 to the negative 4th molar. That gives us a pOH of 3.71, and the pH has now jumped to 10.29. And now we put in excess base of 4 more milliliters. Again, there's no acid to neutralize, so that leaves 0.05 millimoles of sodium hydroxide in 55 milliliters of solution. The pOH of that is 3.04, and the pH has now gone up to 10.96, not quite 11. We then add more excess base, 10 more milliliters. Again, no neutralization going on, so we have 0.15 millimoles of sodium hydroxide in 65 milliliters of solution. Hydroxide concentration is given here, and the pOH is 2.64. The pH is 11.36. In stage 6, we add more excess base. The calculations are very similar, and the pH goes up to 11.52 with a total volume of 75 milliliters. And you can see the characteristic S curve of a pH titration base into acid. This is taken from one of our experimental results. We've got the milliliters of sodium hydroxide added, and up until the point where the acid is gone, we use the acid to calculate the pH. After that, we use the base to calculate the pH. There's a rapid change in pH before and after the equivalence point. Here you can see that on the graph, you can see that in the numbers. So this explains how the pH can make such a vehement increase right around the equivalence point at pH 7. Now an indicator should be chosen to change colors at about the pKa of the salt that's formed in the neutralization. Here, with sodium hydroxide and hydrochloric acid, the salt formed is sodium chloride, and that's going to be a neutral salt. And so we're going to add an indicator that changes colors, right between 6.5 and 8.5, so that that last drop of sodium hydroxide causes the immediate permanent pink color in the solution. So in your titration of hydrochloric acid with sodium hydroxide, phenolphthalein is a good choice because it changes in that area. Here are some other indicators and the pH changes associated with them. You can find these all over the internet. If you're titrating a strong acid into a weak base, which of these indicators would you choose? And I think we all realize that's probably methyl orange, because we're going to change right at 7, and just as the acid goes in, we're going to see a nice change that last drop goes in. We're going to see a change down around 5 or 4. We could also choose methyl red. That might be even a better choice. Some folks would choose bromothymol, and that gets a blue to green to yellow right around the pH of 7.